Our second presentation will be by Dr. Gregory Hageman. He holds a Ph.D. in biology from the University of Southern California, and he's presently a professor of ophthalmology at the University of Iowa. His research addresses mechanisms in the etiology of age-related macular degeneration at a basic science level, and his talk today will address AMD Research.
Well, it's a great pleasure to be here, and I certainly appreciate the invitation from the AMD Alliance International. What I'd like to do for the next 6 or 7 minutes is provide you kind of an update on where we are currently with our understanding of pathways involved in the etiology of macular degeneration. And I have to tell you at the get-go that this is an incredibly exciting time for the field, both for the research field and for the patients with macular degeneration. Just tremendous amounts or tremendous advancements have been made over the last couple of years, and I just thought I'd kind of review how those came to be and where I think we're headed. I do have the following financial relationships to disclose. As David said, macular degeneration is really a progressive deterioration of that region of your retina called the macula, which is responsible for your fine acuity vision. And for reasons we haven't understood until recently, that region of the retina deteriorates with age, resulting in loss of central vision. And as David said, if you're a scientist, the one question becomes how best to try to understand pathways that are associated with this disease. And we know that the earliest stages of the disease are characterized by the deposition of these drusen deposits back in the macular region lying behind the retina. And so these deposits then really become the first clinical sign that you might be destined to develop this particular condition. So we started in this field about 25 years ago with a very simple premise, and that was, could we learn something about what drusen are comprised of, and could we learn something about the origin of drusen, and if we could, would that teach us something about pathways involved in the development of macular degeneration? The primary resource we've used for these many years is a repository of human donor eyes, which really has been the only way to get to some of these questions because of a lack of reproducible animal model of the disease itself. And so the next slide, sad but true, represents about 15 years of research, but we were very surprised to learn what drusen were comprised of. And quite simply, drusen are filled with proteins associated with various inflammatory pathways, and very specifically amongst those proteins are a large number of proteins associated with the so-called complement pathway. And that was a big surprise. It really suggested early on that inflammation of sorts was associated with the primary pathology in the back of the eye, in these drusen deposits. And so let me bore you very quickly. The complement system is part of your immune system, and quite simply, I like to think of the complement system as that first line of defense that your body has to fight against invading microbes, to deal with large amounts of cell death. But it really is that system that kicks in initially, and it works very efficiently. It helps to discriminate between your own tissues and invading tissues, if you will. And I won't go into details, but the system actually is triggered by one of 3 pathways, so you have to have a trigger. We'd certainly like to understand what that trigger is in macular degeneration. And then the system discriminates the good from the bad and in typical fashion tries to get rid of the bad, say a bacterial infection, for example. And it does that by the production of this complex called the membrane attack complex. And that complex quite simply inserts itself into bacterial cell membranes and kills those cells, and ridding yourself of that particular infection. The problem is in various diseases that the complement system doesn't quite function appropriately, resulting in the production of large amounts of this membrane attack complex. And that's really the situation that's happening in AMD. So if you have too much of that membrane attack complex, it actually begins to kill off your normal healthy cells and tissues. And that's the situation in the back of the eye in individuals with AMD. You can see the purple material. Here's a drusen for orientation. That purple material is a section from an eye showing the distribution of this membrane attack complex in an eye from someone with macular degeneration. The real surprise at that point was that not only was there a large amount of membrane attack complex in the back of the eye, probably killing healthy cells and tissues and resulting in that destruction of the macula that I talked about. But the inhibitors, those proteins that are meant to shut the system down at the appropriate time, were present in concentrations very similar to that of membrane attack complex, really suggesting from a scientific perspective that the system, the complement system, was driving to conclusion to the production of large amounts of membrane attack complex in the presence of the major inhibitor. And that major inhibitor is called complement factor H, a very important molecule for controlling this system. And the suggestion at this point in time was perhaps factor H was not inhibiting the system appropriately. That led our group and a number of groups 2 years or 3 years ago now to really look at the complement factor H gene. And surprisingly, we found that variations in the complement factor H gene were strongly associated with risk for developing macular degeneration. So, in other words, mutations in that gene were associated with a large proportion of macular degeneration. And the surprise at the time, if you put a percent to it, it's about 50% of all individuals with macular degeneration have this factor H gene variation associated with the development of the disease. Now, the interesting thing at this point was we found that there is a highly risk form of the gene which is associated with the development of disease. But very importantly, we identified two protective forms of the gene that were strongly associated with protection against developing macular degeneration. And I think that's a very important part of the story. We're also learning that these same variations in the complement factor H gene are associated with a number of additional complement-mediated diseases. So AMD may not exist in solitude with respect to other diseases. Since that time, I think importantly we've learned that a number of genes in the complement system associate very strongly with macular degeneration. And we're now at a place where we can predict AMD with about an 86% reproducibility based on variations in this set of genes. So it's a very exciting time. We didn't really expect that in a very short period of time we would actually understand a great deal about the genetic basis for this disease condition. And I think the wonderful thing for patients who are afflicted with the disease is that there are a huge number of activities out there related to trying to address therapy related to the complement system. And I think we'll find in the next couple of years that we will be able to at least slow the progression of this disease condition based on some of the data that I just showed you. And with that, I'll stop and thank you for your attention, and certainly thank the tremendous number of collaborators that I've had the privilege to work with. Thank you for your attention.